My name's Keith Golding. You can see me on that slide. I'm chairing today. I'm the ECT's Deputy Chair. And I'm very happy to be able to introduce Simon Peacock. I visited Palace Leas a good few years ago now at a British Society of Soil Science summer meeting, and was taken round by Bob Shield, who I think might join us today, although I don't think he's joined yet. So Simon, thank you very much for agreeing to speak to us. As somebody who's worked on park grass for many years and got immense value out of it, I'm well aware of the unique resource that we have in experiments like park grass and Palace Leas. And so we're really looking forward to hearing you speak to us today about Palace Leas. This is the 17th in our series of webinars. I hadn't realised we'd had so many. But it's great to have you on. Can I ask everybody to make sure your videos and your audios are turned off so that we focus on Simon and we don't get any extraneous noises? If you want to ask questions, please use the chat facility. Ben Sykes, our executive officer, will be moderating that and watching the chat and making sure that all the questions are noted, and then he'll introduce you to Simon at the end. But Simon, it's really over to you now. We're looking forward to hearing you. Tell us what's going on at Palace Leas and what opportunities there might be. So Ben will turn it over to Simon now. I'll turn my video and audio off, and it's over to you, Simon. Thank you.
Okay. Can you see my screen?
Yes, I can. Very clear. Thank you.
Right, okay, I've got to find out a way of moving it now. Here we go. Okay, right. Good afternoon, everyone. I hope you enjoyed this snapshot into some of the work that's been done at Palace Leas over quite a long stretch of time. I'll try not to overrun. It's difficult to decide actually what to put in and what to leave out. Okay, the Palace Leas hay plots here at Newcastle University's Cockle Park Farm, about 10 miles or so north of Newcastle city centre, where I'm talking to you from my office in the agriculture building, are the second oldest continuous grassland experiment in Europe, and I believe in the world actually, and they're only the only remaining survivor of lots of original experiments set up in 1897. Other grassland experiments across Europe have been heavily influenced by experimental shifts and many changes in methods over the years. Palace Leas has had, with one or two minor changes, the same continuous management for 124 years, effectively the same experimental plots and the same soil amendments. The only change of any significance was changing over from the basic slag which supplied phosphorus to a superphosphate because of the chemical content of the basic slag was becoming unreliable. Today, the plots, as they were over 120 years ago, are cut in late summer for hay. This cut is subsampled and measured for forage quality. The aftermath is then grazed and the soil amendments are added every spring. It was originally set up to determine how difficult soil amendments and combinations of them affected hay yield and quality. On this picture, you can see the rather tatty signage that's been there for a number of years, which we are hoping to update in the near future. Okay, here's a plan drawn about 1902, showing the location of the Palace Leas plots in the top left. I think you can just make out the rather oddly shaped trapezoid plots numbering 10 to 20 in the top right. The plots after 13 were removed fairly shortly after the start of the experiment. Incidentally, the layout of the farm hasn't really changed all that much today, and most of the field you can see in the map is still in existence. Okay. This is a more contemporary aerial view of the plots, and you can easily see how the different treatments influence the composition of the grassland, easily spotted by the different tones of green. This table gives a breakdown of all the research topics that have been carried out at Palace Leas over the past 100 years or so, but of course most of the last 30 or 40 years. This is a plan of the experimental plots. As you can see, the shape of the plots is rather governed by the shape of the field. It's a sort of lozengey shape. So we have 14 plots and a guard plot between plot number 1 and the road. This buffer zone you can see next to the road has been mistaken for plot 1 in the past by visiting scientists, which means they've compromised their sampling machine. But I can assure you, not in the work we'll look at today. The main reason for this is the plots are separated only by white poles at the plot boundaries, and when you're in the middle of the field, it's notoriously difficult to assess exactly which plot you're in. The table on the left shows the amendments that have and still are applied every spring. We have FYM applied solely and in combinations with mineral fertilizers in the first 5 plots. Plot 3, 4 and 5 have FYM applied every alternate year or every 4 years. It mostly comes from the farm at Cockle Park. Plot 6 is a control which has had no amendment since 1897. Plot 7 is a mineral nitrogen only plot, and the soil acidification that this has caused over the years has produced a distinctive, quite deep peaty layer on the surface, which is devoid of all soil fauna and is very, very acidic and plant species impoverished. Plots 8 to 13 have various combinations of mineral fertilizer, individually and collectively, and plot 14 represents a contemporary mineral fertilizer treatment. All the others reflect historic applications which are no longer in use, but it's felt that these should be retained to preserve the integrity of the experiment. So this is a table of the soil amendments as presented in the book Cockle Park. This was published in 1959, giving a historical perspective. For those who aren't used to these, muriate of potash is potassium chloride, nitrate of soda is sodium nitrate, and slag is a byproduct of steel making and is rich in phosphorus and lime. So effectively, pre-1976, plots also had lime added to them. It's quite an old archaic way of presenting these results, but I thought you'd be interested in seeing some of the older stuff that they presented. This is a rather quaint way of presenting results of hay yields of the plots. The shaded areas being the yield. These are the averages from 1897 to 1902. Already differences can be seen, and we're even provided with a selling price in pounds, shillings and pence. Okay, a slightly more modern approach. The yields printed graphically for the first 10 years. The FYM plot 2 shows the highest yield here, whereas the lowest yield is from the control plot. Okay, and you can see at the background on the right, that's an aerial photograph of the Palace Leas plots. Okay, a more contemporary table showing average yields between the start and 1984 here in kilograms per hectare. Differences in yield are very different between plots, the lowest being in the control and plot 9 receiving only potassium. However, these lower yield plots have the highest crude protein levels and metabolizable energy. This one here is from a paper in PLOS headed by Jonathan Kidd, who was a PhD student in Newcastle a few years ago. And there's an assemblage of Janet Simkin, who's in this seminar, Liz Stockdale, myself and Pete Manning. We have contemporary yields from between 1976 and 2005 in graph A, top left, again showing the lowest yields in the control and the potassium and the nitrogen and potassium, P limiting plots, and the highest in the farmyard manure plots. Graph B, top right, shows grass cover as a percentage. There's no real significant differences here except the control and the phosphorus amended plots. Graph C, bottom left, shows the herb coverage with the phosphorus amended plot showing a significantly different, almost 60% cover. Interestingly, the N and NK plots, which have no phosphorus amended to them, have no herb cover at all. In C, we have legume cover. Sorry, in C, we have herb cover. Again, nothing in the NNNK plots, and curiously, very little in the FYM plots either. These graphs indicate that the NNNK plots are entirely grass covered. Jonathan also compared contemporary hay plots with the historic first 5 years, from 1897 to 1901. And I think it's obvious that apart from plot 2, the farmyard manure only plot, that the yields are very comparable between 120 years ago and relatively recently, about 10-20 years ago. Okay, here we come to the aftermath grazing and work done, I think this is by Steve Pullen a few years ago. This is a plot I found in the archives and shows the preference of stock on selected plots. This was work carried out in the 80s, I believe, by the use of exclusion cages. He's demonstrated that was a preferential grazing of the control and phosphate only plots at the expense of the more unpalatable nitrogen only plots. Possibly this is related to the herb coverage or lack of herb coverage. So you can see, if you look at the nitrogen plots, the proportion of the regrowth is only 50%, whereas with the phosphate and the farmyard manure plots, we're up to about 75% of the proportion that's the regrowth that's eaten. Okay, so David Hopkins, who worked with Robert Shield actually for a number of years, did a much cited study on carbon stocks at Palace Leas. He investigated carbon stocks in 2006 and compared them to a similar study done in 1982. These graphs show, not very clearly I'm afraid, that the carbon stocks at Palace Leas have changed very little over a period of 2 to 3 decades, including, surprisingly, the very peaty and acidic horizon plot 7, which receives nitrogen only. I would say this suggests this horizon and the acidification of plot 7 started quite a while ago, so I don't know how this could be reinterpreted as we put more emphasis on carbon budgets in future years. Okay, this is part of the same study and shows even more dramatically that little has changed when looking at the soil organic carbon stocks as a function of depth between 1982 and 2006. Jonathan Kidd, incidentally, also looked at soil organic carbon contents more recently in 2015, I believe, and his results seem rather different to David Hopkins. He's not surprisingly found high concentrations in the farmyard manure amended soils, but he doesn't seem to have picked up the high concentrations found by David Hopkins' study. So this shows you that even researchers doing a similar thing can often find quite different results. Okay, looking at more results from Jonathan Kidd's study, graph A plots the pH of the top 5 centimeters of the soil profile. We can see relatively high pH values of the farmyard manure amended plots, but the nitrogen amended, the nitrogen and potassium amended, and the contemporary NPK plots are very acidic, below pH 4, and this acidity is reflected in the plant species cover, as we'll see. pH of the soil between 5 and 10 centimeters, top right, shows less extreme values. The extractable phosphorus is high on the farmyard manure plots and also on the phosphorus amended soils, as you would expect, and is nearly absent on the control plot and the mineral N amended plot. That's graph C. And there's a similar pattern with the exchangeable potassium, which is graph E. The concentrations found in the potassium amended plots tend to be the highest, as one would expect. But there's only minor differences between those not amended with potassium and the farmyard manure amended plots. Okay, we have a paper by Thomas, Holmes and Clapperton in who did a chemical analysis on the pooled vegetation biomass of each of the plots in the early 50s. And they presented a paper, and it's in the Empire Journal of Experimental Agriculture. I don't think that's running anymore now, in 1955. They use plant material from all 14 plots, and in the 50s they must have had to have analyzed it using wet chemistry techniques. Today, of course, we've got atomic absorption, ICP analysis, much quicker and much easier, of course. Myself and Robert Shield, we had 2 postgraduate students from Brazil doing summer placements in 2014. I think that was the summer when Brazil were spectacularly eliminated from the World Cup. So we were lucky that they finished their work, actually, because they were devastated halfway through the study. Here are their results. Excuse me. And then they're compared with Thomas, Holmes, and Clapperton's from 1955. I'm not sure how well these can be interpreted, possibly as just a snapshot, because obviously techniques are different, not necessarily more precise, and concentrations may have possibly changed over a period of 60 years. Top right, we've got the calcium, and it compares content in percentage, and it compares very well. Perhaps the amount measured is slightly lower than in 1955. The coloured parts of the plots, incidentally, are the contemporary results, and the plot outlines in shadow are the ones found by Thomas, Holmes, and Clapperton. Okay. On the other hand, magnesium, bottom right, the levels are slightly higher, as are the potassium ones as well. So they all seem to compare very well with the historic values that were found in the vegetation. This one here shows the percentage of sodium, and we've got elevated sodium levels. But with the exception of plots 8 and 10, the phosphorus amended plots, there's a remarkable fit, I think, between the historic amount and the amount we found in 2015. We have trace elements as well, copper and cobalt, very important for nutrition. I think, again, they show a remarkable fit between the original results found in 1955. And finally, we have iron and manganese. Again, a good fit against the 1955 study. I think further work here would be of great interest, and I think we're going to intend to do it over the next few years, including important mammalian trace elements such as nickel and selenium to determine how soil amendments affect the availability of these vital trace elements. A study of the soil component to complement it would also be very useful, I think. We also come to plant species composition. This is a table of simplified botanical composition of selected plots. 2 with farmyard manure, 6 control, 7 nitrogen only, 8 with phosphorus only, 10 with nitrogen and phosphorus, and 13 with NPK. This is from the archives, and I'm struggling to find its origin, unfortunately. It's dated 1971. Here we have Lolium dominating plot 2, the FYM plot. There's Festuca dominant in plot 6 and 10, the NP plot. Today, I believe Festuca is dominant in plot 7, the acidified nitrogen-only plot. This gives a very historic snapshot of the composition of the swards of the plots at Palace Leas about 50 years ago. Okay, here we have a contemporary botanical survey carried out in 2014. There have been only a dozen or so of these botanical composition studies done over the last 100 years. One was carried out in 2016, and we hope to carry out another one over the next year or so. You can appreciate the timing in June is bad for us teaching academics, because these are recorded using the usual NVC methods carried out by Janet Simkin, myself, and Helen Adamson here at Newcastle University, and analysed by Janet. Just to show you these results. So we're looking at preferential species grasses here on the plots first. These are some very condensed results. Top left, we have Agrostis capillaris, abundant in all the mineral fertilizer plots, but also almost completely absent in the farmyard manure plots. The top right, Holcus, appears to be performing the opposite. Very much present in the FYM plots, but not in the mineral fertilized or the control. The middle left, Olopicurus foxtail, seems to have a similar pattern to Holcus, whilst Festica is very similar to Agrostis, favouring the mineral amended plots. Anthoxanthum is present almost throughout the plots, so there's a very different distribution of grasses between the plots. Next one is the forbes. I would say the forbes appear to favour the mineral fertilizer amended plots. Anthriscus, the cow parsley, is only found in the farmyard manure plots and nowhere else. I'm tempted to say that the seeds were present in the farmyard manure when it was brought in, but judging by the cow parsley present outside, I don't think that's the case. We have Rhinanthus minor, hay rattle, only present in the control and the peat amended, potassium amended plots. Okay. So here we have species richness and diversity. Species richness is highest the control and the phosphorus amended plot 8, as is the Forbes species richness. It's lowest in the acidified plot and to a lesser extent in the other mineral fertilizer plots. We've also picked up some mosses in plots 9 and 11, the K amended and the NK amended plots. The N2 hills diversity, otherwise known, I believe, as the effective species diversity, is again highest in the control plot and the P and PK amended plots, and surprisingly lowest in plot 1. This is the NBC analysis, showing the entire grassland as it was at the start of the experiment to be an acid grassland, a UB4, a Festuca agrostis community. It shows over time, particularly in the FYM amended plots, how the communities have changed over 100 years or so and become more like MG7D, which is a lowly Malapicurus community, or an MG5A, the Cynosurus centuria grassland community. Okay, this is from a paper published in 2016 by Roy Sanderson and a PhD student of his, Karen Evans. She was looking at invertebrates, specifically arthropods at Palace Leas. She looked at two types, those collected by above ground by sweep nets and the more below ground pitfall traps. This is a component analysis you see here. The results demonstrate that soil nitrogen and FYM are important determinants of arthropod community composition. And you can see the cluster of these herbivores and parasites and pollinators around the center of the principal component analysis there, compared to the treatments on the right principal component analysis. The second graph shows redundancy analysis from the pitfall traps and shows that the soil acidity influences the arthropod community very well. And again, you can see the clusters of the pollinators, the parasites, and the predators crossing around the controls and the FYM on the right-hand side of the plot. Okay, if it's possible, if you look carefully down the experiment on certain days at Palace Leas and the vegetation heights, you can still see the old ridge and furrow arrangement as a result of the ploughing carried out at Palace Leas in the late 18th and early 19th century before the experiment was set up. This is a short archive study carried out, I believe, in 2007 by James Taylor, who's left us now and is at the Ecological Institute in France. And it shows the difference in pH between the ridge and furrows on plot 6, the control, and plot 7, which is the nitrogen mineral nitrogen only plot. You can see on plot 7 how the pH appears to indicate acidification of the soil in the ridges, but not in the control plot. I think maybe this is a study we need to take further as well. Okay, there's also archive material found on the swall height, differences between the ridge and furrows in 2007. And you can see here that on all the plots, the vegetation on the furrows is consistently higher than the vegetation found on the ridges. And finally, another piece of work that needs taking forward from the archives. This is a GPS map of the fungal fruiting bodies, as I say, data collected by GPS. And you can see the abundance on the FYM plots compared to the rather fungal, barren, acidified plot 7, for example. And again, this is another study that we should be moving forward. Okay, that's me done. That's me done for this. I hope you've enjoyed this rather whistle-stop tour of our Palace Leas experiment at Newcastle University. Thank you all very much for attending.